Thank you, Miranda. And hello, everyone. My name is Emmy. I'm the account manager at Novogene Europe, specifically in Finland, and I will be the host of this webinar. I'm very delighted to see that so many of you were able to join us today. Today, my colleague and Deputy Product Director, Dr. Chiki An, will go through next generation essentials for 2024. But before that, let me briefly introduce our company. Novogene was founded in 2011 and has been growing rapidly since then. We're continuously developing our portfolio of NGS and third-generation sequencing, and today we are present with services in various fields of science, from research, oncology, to food water testing. Novogene is a global company with local contact persons and sequencing facilities in Europe, Asia, and US. We have over 2,600 employees and more than 5,600 customers. Our multiple sequencing facilities equipped with Illumina, PacBio, and Oxford Nanopore technology gives us the largest sequencing capacity in the world. Let's have a look at specifically Novogene in Europe. So our first European sequencing center is located in the UK, in the Cambridge Science Park. This state-of-the-art lab is equipped with Illumina technology and our patented intelligent sequencing platform called Falcon 2. The automated workflow of Falcon enables us to decrease turnaround time, minimize the factor of human error, and by doing so, yielding consistently high-quality outcomes. Thanks to our supercomputing capacity, we can also process large amounts of data. As you might have noticed, we have recently launched another sequencing facility in Europe, specifically Germany. The German facility is gradually becoming more and more operational, so stay in touch with your local contact person to receive the latest updates relevant for your upcoming project and the German lab facility. We provide a full range of next-generation sequencing services. This can be human, plant, or animal DNA sequencing, various types of RNA sequencing, metagenomic services, and epigenomic services. In reality, our service offers extend even beyond what is mentioned on this slide. We offer, for example, oncogene panels designed for solid tumors or hereditary cancers, and also accept pre-made libraries for sequencing. So why Novogene? There are many reasons why you should choose Novogene as your sequencing partner. Providing consistently high-quality services is our priority. And to achieve this, we perform multiple quality controls or QCs. There's sample QC, library QC, and finally data QC. We have a large capacity, which we are increasing all of the time. Recent examples of this would be opening our German lab facility and also investing in the newest sequencing technology with improved throughput. Our patented Falcon 2 technology has not only a positive impact on quality and capacity, but also on turnaround time, since Falcon can run 24/7. We offer customizable end-to-end solutions. This means we can process your project from extraction to final analyzed data, but you can also send us already extracted DNA and RNA or pre-made libraries and choose to only receive the raw data. Our product management team and laboratories are continuously optimizing protocols and services to ensure that we have a sequencing solution that fits your needs. We are very proud to support researchers worldwide in their work, and today have a clear record in the scientific community. We have been cited in over 16,000 research papers, with a total impact factor of over 10,000. We are also driving the NGS industry forward with 69 NGS-related patents. Again, zooming in on Novogene in Europe. Your contact persons are present in almost every European country, So get in touch with your local representative today to discuss NGS solutions for your project, either by contacting them directly or via our websites. We also regularly host seminars in universities and various institutes across Europe. So if you would like us to organize a seminar near to you, don't hesitate to be in touch to discuss this further. At this point, I would like to briefly introduce to you the faces of our talented European sales team. They are based in different regions across Europe and functions as the first person of contact to Novogene. So keep a lookout for your region. Here on this slide, we have UK and Ireland, as well as DACH, so Germany and Austria. Here we have Southern Europe, Nordics, Eastern Europe, and Benelux. And with this being said, we speak your language, so do not hesitate to contact us with your next generation sequencing related request. Thank you for listening. That's all from my side for now. And Shichi, over to you.
Hi, and Ami, and thank you very much for the introduction. And I have my shared slides and get things set ready. Okay. I'm just going to put them in present mode. Okay. Just 1 sec. And then I'm just going to have my point out, and now we're ready to go. Okay, thank you again for the introduction. And without further ado, I'm going to go into the main content, what we're going to cover today. So here's a quick overview. I'm going to start with a little bit of introduction about what is actually DNA sequencing. And then we'll discuss about some sequencing platforms that are available, including Illumina, PacBio, and Oxford Nanopore technologies. And then importantly, we're going to have a quick comparison about their features and also help you to select what would be the most suitable platform to use for a project. And then we're going to talk a little bit about the most common DNA sequencing approaches, including whole genome sequencing, whole exome sequencing, and target region sequencing. And through those approaches, one of the major results is identifying mutations or variants. And we're going to dive into a little bit more deep regarding how those information are important in human genomic research. And we'll talk a little bit in cancer research and also in disease genomics as well. And of course, DNA sequencing applications go far beyond just sequence genome, and there's some other applications there we're going to touch on a little bit today as well. And in the end, I'm going to briefly tell you what is to expect for Novogene Europe in 2024. So please stay tuned. Okay. And so we're going to start with about what is DNA sequencing. I think most of the audience here probably know as basically a process of determining the nucleic acid sequence, the order of the nucleotides in the DNA. So this technology has broadly used in life science and also medical field as well. So this is actually growing really its popularity and really revolutionized the life science research basically. And still, researchers continue to push the limits of current technology to be able to accommodate more challenging samples and develop more applications as well. So here's some of the species that actually are sequenced by Novogene got published in collaboration with our customers as well. And so we can see it covers a really broad range of species from human, different animals, plants as well, and also microbial genomes as well. It really have a broad application. Okay, and now, so since this is really, really important, has broader applications, now I'm going to spend a little time about the tool in sequencing. So generally speaking, there are 3 generations. The first generation is also known as Sanger sequencing. That is still probably broadly used for example, cloning or validation of small fragments as well. So this is relatively expensive, and then the sequencing length can only take on probably around 100 BP fragment. So it's considered as a short read sequencing. And the second generation sequencing, also known as high throughput sequencing, and then the most widely used platforms are Illumina platforms. So this is the platform technology that revolutionized the sequencing field, basically, and is able to conduct high throughput sequencing. Millions of reactions can happening in parallel. And this is fast and more affordable, but this is still considered as a shorter read sequencing as well. So it's only able to sequence fragments between around 50 to 100 BP fragments. And more recently, there was a development of the third generation sequencer. So this is featuring a long read sequencing. And so again, the most commonly used platforms are Oxford Nanopore platforms and PacBio. As you can see here. Both platforms are able to sequence native DNA in real time with a single molecule resolution. Okay? And both of them are able to sequence tens of kb fragments on average. So in the next few slides, I'm going to dive into a little bit more about the sequencing principles behind each different platforms. Okay? So I'm going to start with Illumina. So what is the technology used behind Illumina sequencer? It's sequencing by synthesis, or SBS. How this generally works. So basically, in each sequencing cycle, one of the fluorescently labeled nucleotide will be added to the growing nucleotide chain. So the polymerase is going to add them into the new chain. Okay? And then the fluorescent basically then will be read. Okay? And the image will be captured, and this signal will be translated into the nucleotide acid sequence. So that's how we know what is being sequenced. Okay, and then it actually contains a terminator. So once the first round is finished, and the terminator will be removed, which will allow the next cycle to start, and a new nucleotide acid will be added. It start a new cycle, and this is how I actually gain information of the DNA sequence. And something I do wanted to mention here is that late last year, Illumina just launched their newest flagship sequencer known as Novogene X. So this platform contains a lot of innovation technologies. I won't go through them all. Just a couple of things I would like to highlight. One of them is ultra high density flow cell, which enabled high throughput sequencing even further. And then also incorporated the new chemistry. It's called an SLEEP SBS. So this also optimized shipping and also improved sequencing as well. Okay, so here I'm just going to use some parameters to show how this new platform actually further enabled broader and deeper sequencing. So here, what I listed here is Nova 6000, and I use S4 flow cell example here. And for X plus, and I have a 10B flow cell and 25B flow cell listed here for comparison. So you can see here the lane numbers. S4 only contains 4 lanes, and for flow cell on X plus, it contains 8 lanes. For the chemistry, they both use 2-channel and SBS chemistry. It's just the signal capturing wavelength is slightly different. Okay. Something I would like to highlight is that the data output. You can see here. So per lane for a 6000 S4 flow cell, it's generate about 750 GB of data. And then for the X plus 10B, it generates around 375 GB per lane. So although it looks like it is less data per lane, but because it has 8 lanes, you can see the total data amount generated is really comparable to 6000. And importantly, because per lane it generates slightly less data and gives more flexibility if you're interested in lane sequencing for a smaller project. So you can have the whole lane very easy and cost effective to yourself, to your own project. And then 25B flow cell, this is the one basically a game changer. So you can see per lane is able to generate 1 TB of data. Okay, and it has 8 lanes. That means it can generate 8 TB of data per lane. And in each run, actually, we can run 2 flow cell in parallel. So that means per run is actually able to generate 16 TB of data. So you can see the throughput is significantly improved, which also means made the sequencing more cost effective even further. Okay, and currently actually class-based services are now available both at our Novogene UK and German laboratories. Okay. So next, I'm going to move on to PacBio sequencing. And so the technology behind PacBio platforms is single molecule real-time sequencing, so smart sequencing. And this is basically the smart cell, how it looks like. Also see from here. So the size is about a fingernail size. Okay, so within the smart cell, it contains millions of wells. Basically, it's called zero mode waveguides. You can see here. Okay? And then you have your library molecule generated. Okay. Okay, so this is how it looks like as a loop. I'm going to come back to this point later. The circular template, okay, will be sequenced. So this molecule basically will be immobilized at the bottom of the ZMWs. And then during the sequencing, the polymerase will also add fluorescent labeled nucleic acid into the new chain. And then the signal then will be captured. And by reading those fluorescent signal, we know which base was actually it's incorporated in the new chain, and therefore get information of the DNA sequence. Okay, this is how generally works. So now I'm going to talk a little bit more about the sequencing mode of PacBio. So actually it enables two different sequencing modes. One of them is called a continuous long read, CLR. Another one is called a circular consensus sequencing, CCS. So what's the difference? So for CLR mode, it allowed a slightly larger insert size to be sequenced, but the polymerase is only able to sequence this molecule once. The insert once. So, however, under this mode, okay, and the error rate is still relatively high. Then on the other mode, CCS, it sequences a smaller insert, but the polymerase is able to sequence this fragment multiple times. So because the errors are random, if you sequence the same fragment multiple times, you can use the information to correct itself and then generate a high fidelity read, also known as the hi-fi sequencing, hi-fi read. Okay, so this is quite unique to PacBio platforms. And also later last year, PacBio launched their newest sequencer, and that was Revio. So here, again, a quick comparison between the previous Sequel2E system versus Revio to show you how the new platform unleashed the power for hi-fi sequencing. So from the smart cell, you can see the previous smart cell contains 8 million ZMWs, and then the new Revio smart cell contains 25 million ZMWs. This, you can see 3 times increase of the capacity already here. And on the run mode, the older version able to run both the legacy mode CLR and also the hi-fi mode CCS. Okay. And Revio is optimized for run hi-fi sequencing. You can see the hi-fi data yield per cell. You can see a clear significant increase under the Revio system. Okay. And importantly, Revio system is able to run 4 smart cells in parallel. So this further increase the capacity, again, making it more cost effective when sequencing hi-fi data. Okay. And also, I do wanted to mention that for PacBio, sequencing is able to also detect methylation in real time as well. When you sequence the DNA, it doesn't require a different type of library preparation. Okay. And now we're going to move on to Oxford Nanopore technology. So this is very different. Okay, how this works. So nanopore technology basically it's nanometer sized pore, protein pores formed on membrane. So during the sequencing, the DNA will be unzipped. One of the chains going passing through the pore. Okay, when the chain passes through the pore, it's going to cause a current disruption. Okay, and each base basically causes a different type of reduction at different level. So by capturing and the disruption of the current, then we go to understand what is the base actually passing the pore, and therefore decipher the sequence of the DNA. So that's measuring something quite different from the other two platforms. So what is new for nanopore technology? And so they recently launched the Q20 plus chemistry for single molecule accuracy of 99% and higher. So for this newest Q20 plus platform, what it contains, it has a new pore. It's R10.4.1. It has improved pore design to fine-tune enzymes, enables faster speed, and helps improve the Q score, the accuracy as well. You can see it's much faster, and then the accuracy has improved as well. It also contains a new motor, and so it's with improved movement properties as well. So for Novogene, we already adopted this new kit, basically including a version 14 kit chemistry and plus the new pore. So for our customers, we're able to offering this new technology, new update to all customers. Okay, so here is a quick summary about the principles that behind the different platforms. So for Illumina, it's a sequence by synthesis. So basically, I read fluorescent after each letter is added, and then remove the fluorophore, allow the next one to be added. Okay. And then for PacBio, the technology is called single molecule real-time sequencing, smart sequencing. It is also read fluorescent as the letter is added, but this is a real long read sequencing. It's real time. It's like in taking a movie when the new chain is synthesized. Okay. And then for Oxford Nanopore, it measures the changes in current when the letters or the base passing the pore. So it's measuring something quite different compared to the other two platforms. And so as promised, here's a quick comparison across different platforms. Okay. And so for Illumina platform, this is a short read. This is still really broadly, widely used in NGS industry. A lot of applications are developed based on this platform, and offers the lowest per GB sequencing cost range, okay, and has the highest confirmed data output. Okay. And also the error rate is quite low. So it's really a robust, reliable platform for sequencing. So the limitations is very obvious. Okay, the read is very short. And then it has limitations when it comes the applications that require de novo assembly, basically, or assembly in general. Okay. And then for PacBio and Nanopore, they shares a lot of pros and cons. For the pros, both of them are real long reads. Okay. And both of them are able to direct detection of epigenomic modifications such as methylation. Okay. Both of them generate real-time data output. Okay. So PacBio has another advantage is that they under CCS mode is able to generate extremely high accuracy data. Okay. And then for Nanopore, it's able to generate probably even longer reads compared to PacBio platform. So they also share actually a lot of counts together. Both of them are more expensive compared to Illumina sequencing. And then both of them requires a more large amount of starting material and high quality DNA and RNA for library preparation. And so the sample quality is very important if you want to use both platforms. And both of them suffers a higher error rate. So error rate for PacBio, if it's under CR mode, the error rate is still high. And then if you choose CCS mode, it has a higher accuracy, but the read will be shorter. Okay. And so for Nanopore, so it is quite, because for the biological pores, it's quite sensitive to environment changes. And so they share some very common cons here. Okay. Now we have talked about the platforms, and now we're going to move into some general approaches that are broadly used in RNA-seq. Okay. So I'm going to start with the most common one, the whole genome sequencing, WGS. So research performed this is basically want to obtain a comprehensive pictures of an organism's genome. And by the experiment settings, you have two basically different designs of experiments. One of them is called de novo sequencing, another is resequencing. So for de novo sequencing, basically, you wanted to sequence a novel genome when there is no reference genome available for a line tumor to map to. What you wanted to achieve here is that to assemble and annotate a reference genome. Okay? And then for resequencing, it means sequencing a new example of a species and which you already have a reference genome. So what you want to do here is that to compare differences between genomes from the same species. So you want to identify variants or mutations, basically. And if you want to do a resequencing. And so here's another way to look at the difference here. So the top panel here is for resequencing. So basically you have your reads and then you have a reference genome. Basically you aligned reads to reference genome to identify variants. Okay. And then for de novo, you don't have a reference genome there. So you basically will need to construct a genome sequencing from those overlapped reads. And this is a assembly-based approach. Okay. This is a mapping, this is assembly-based. Okay, so this is a major difference. And then for resequencing, as I mentioned, one of the main aim is to covariance, basically. Generally speaking, whole genome sequencing will allow you to identify all different types of variants. For example, single nucleotide variants, basically one base and change, and also deletions, insertions, or together they can be called indels, but they're generally small ones, less than 50 pp. And anything bigger than that can be grouped as structural variants. Okay, for example, copy number variants or duplications, translocations can also be identified through whole genome sequencing. Okay. So next, I'm going to talk about whole exome sequencing. So from the name, you probably know this approach basically focuses on the coding region of the genome. Okay. So why researchers wanted to study or only sequence the coding region? Here's some reasons here. So for example, human exome represents less than 2% of the genome, but actually contains about 85% of the known disease-related variants. Okay, so which means if you only sequence the 2% of the coding region, you actually gain a lot of information of those disease-related variants here. Okay. That means with the same sequence data amount, you're going to have higher coverage, which will help you to identify rare and low frequency mutations as well. So in another way, it can be considered as a cost-effective alternative to whole genome sequencing. Okay. And so there are a lot of panels that can be selected from, okay, but generally speaking, it always consists of two major steps in whole exome sequencing. So the first step is an enrichment step, select only a subset of DNA that encodes protein. So basically enrich exome here. And then the next step is the sequencing step. Basically use the high throughput sequencing platforms to sequence what you enriched from the first step. Okay. So again, as I mentioned, there's a lot of panels and probes can be used, and we support at Novogene, we support our different types of panels as well. The ones that I wanted to spend a little bit more time on is the newest launched panel twist exome 2.0. So what makes it different, stand out, is because it's focused on functional content and also clinical relevant targets. So this is a really well-designed panel. It's designed in human HG38, okay, and it includes content from coverage of specific regions and also PGX important SNPs. You can see it include coding content, and also including content that with additional evidence, okay? So potentially important as well. And then also non-coding regions and some other content. For example, that all pathogenic variants identified in the CLINOA database as well. Okay. So, and importantly, this panel is optimized for NovaSeq instrument. And then the enrichment is very uniform, and all the NGS all the probes NGS-QC as well. Okay. So now next we're going to move on to the targeted region sequencing. Again, here for this approach, basically it's more focused on only a select of genes or regions that are known or suspected to associated with a disease or phenotype under study. This is even more focused than the whole exome. Okay. So generally speaking, there are two major methods to enrich the target region. So one is a hybridization capture, which in a way is very similar to whole exome sequencing approach. It's just the design, the probes only covers a select of genes or regions. Okay. And again, at Novogene, we have our own clinical-based panel for Nova PM 2.0. So it's a panel for cancer research. And then we also support Agilent design probes as well. And then for mitochondrial genome, we actually have a Roche-based panel for sequencing mitochondrial DNA. I'll come back to that later. And there's also other ways to enrich as well, that is through PCR or amplification. Okay? Those can also be achieved through amplification and sequence amplicon, and then to focus on target genes or regions. So here is a quick comparison across the three different approaches. For whole genome sequencing, in principle, it's able to cover all genes, including non-coding DNAs, identify all types of mutations. Okay, and then if you want to reach the same coverage, okay, so this obviously is more expensive. That means you have to sequence all the whole genome and require a larger data set to be sequenced. Okay, and for whole exome sequencing, okay, it only covers the entire whole exome, and many mutations can be identified and tested, but it's only in the coding region. Okay. So for the same data amount, obviously this is going to give you higher coverage, which made it more cost-effective. And so for target region sequencing, this only covers a very small number of genes or regions, and then only a small number of mutations obviously can be identified. And so for the same data amount, sequence data amount, obviously this is going to give you even further coverage. So in that sense, it is probably most cost-effective. And so saying that, there's something I do want to highlight. So given the sequencing power capacity has been increased, and also sequencing per GB pricing has been dropping, and so WGS becomes quite trendy, and because whole genome sequencing is basically the one provided the most comprehensive analysis of variant types. Some other of the approaches have limitations on variant detections. Okay, just something to bear in mind, okay, when you design your experiment. Okay. So now I will talk a lot about sequencing and variant detection. I'm going to use human genomic research as an example to show why this is important for human genomic research, okay, in the next part. So I'm going to start with cancer genomics. Okay. And so every cancer genome basically is uniquely altered from its own host genome. Okay. So what this means, so you probably have heard this quotation from this very famous author. So all happy families are alike. Each unhappy family is unhappy in its own way. In a way, a cancer genomics can be very similar to this. Basically, normal human genomes are all alike or mostly alike, and every cancer genome is abnormal in its own way. Okay. So therefore, it's highlighting, it's really important to identify those variants. And then also each cancer genome has a unique set of genome alterations from its normal host. So basically, this is what it means. I'm going to go into a little more details in the next few slides. So first, I'm going to introduce some terminology here. I probably heard a lot in cancer research. So the first one is the germline mutations and somatic mutations. So what is the difference and why we want to identify somatic mutations in cancer research? So for germline mutations, it's basically any detectable and heritable changes in germline lineage. So the mutation actually happens at the parental gametes level. Okay? So it's inheritable to the offspring, and this is also known as hereditary mutations. It's going to impact the whole organism. And then on the other hand, we have somatic mutations. Basically, this is mutations happened in somatic tissue, okay? And it will result in genetically mosaic individuals. It's only impact part of the individual, not the whole individual. Okay? And also it happens at any time of the lifetime, except the parental gamete stage. Okay? So this is not inheritable to offspring, and also known as acquired mutations. Okay? So why it's important to study somatic mutation? Because the most common cause for cancer is actually somatic mutation, and germline is far less common. Okay? And then what it consider why it is important to understand the difference is because the identification of somatic mutation actually requires parallel sequencing data that directly comparing the DNA sequencing from the tumor sample and also their normal counterparts. So basically, if you want to cause somatic mutation, you will have to have a paired design which contains tumor samples and also the normal counterparts in a paired sample in order to cause somatic mutations. Okay. And then another concept in cancer research I wanted to mention or introduce to you is driver mutations. Okay. So understanding driver mutations is really important for us to understand the genetic changes of cancer. So what is driver mutations? Okay. Basically, those mutations are the ones that provide a selective growth advantage and promote cancer development. Okay, and termed as a driver mutation. It's like a driver sits in the car, okay, is able to decide which direction the car goes, also how the cancer develops. Okay, that's why it's called driver mutation. And then once it doesn't have impact, it's called a passenger mutation. They just tag along in a way, like a passenger in the car. They can't decide where the car goes, basically. Okay, so here, just want to highlight why this is important. So basically, cancer cells will accumulate somatic alterations over time. You can see from fertilizer-like, also under different environment selections as well, accumulate somatic mutations, including driver mutations as well. Okay. And then also mutation frequency depends on the cancer types as well. Okay. And so another aspect variants can be used as to help us understand the tumor evolution and heterogeneity. Okay? So this variants information can really help us to understand tumor heterogeneity and also the shape of the tumor microenvironment over the course of treatment regimes. Heterogeneity is very important in cancer research because this is the one that affects the key cancer pathways and also driving phenotypic variations as well. They all root from the heterogeneity of the tumor. And this also poses a significant challenge to personalized cancer medicine as well. And so that's why I understand the variants or mutations that happened in cancer can really help us in understanding cancer and developing treatment as well. Okay. And so another quite important concept here is tumor mutation burden. Okay? So this becomes a very important clinical biomarkers that associated with cancer response, basically to immune checkpoint therapy. So what is tumor mutation burden, TMB? Okay? It's basically referred to the number of genetic changes, mutations in a cancer cell, and it's reported as mutations per megabase. Okay. So how does it work? So basically, if you have a DNA change, okay, usually will result in amino acid change in the protein, and a new protein change will result in a new shape. Formally it's called a neoantigen. Okay, so that is actually on the cell surface as a marker that is actually important for recognition by the immune system. Okay? So basically, when you have a low mutation burden, you have fewer mutations, and then this actually decreases the chance for one of them get recognized by the immune system or activate the immune system. So that means the response to immune checkpoint inhibitor therapy will be less, okay, will be weaker. And then cancer with a higher tumor burden, okay, so it means they have more mutations and also increase the chance that at least one of the marker can be identified by the immune system and activate immune response, which means they probably have better response to the check inhibitor therapy, immune checkpoint inhibitor therapy. Okay. So just some important things in cancer research and that related to variant detection here. Okay, now for the next bit, I'm just going to spend a little bit of time on disease genomics and why identification of variants is important in here as well. So for genetic disease, actually, it falls on a very broad spectrum of genetic architecture. So many people, when think about genetic disease, as more thinking about rare disease or severe monogenetic disease. Okay, so they are probably the cause variants are typically Mendelian. Okay, they're monogenic, but it has a big impact. Okay, but actually, it also variants also contribute to common complex diseases as well. So they're generally massively polygenic, okay, usually caused by a combination of many genes and also environmental factors. And the cause variants tend to be more common and with small impact basically. So just a little bit in rare disease first. So why variants calling is important, identification is important. So actually there are more than 7,000 known rare disease and more to be discovered every year. And less than half of them actually have resolved a genetic etiology. And it's estimated there are 300 million people suffering from rare disease. They impact more people than AIDS and cancer combined. So when you think about rare disease, but actually it's not a real rare, it impacts a large population of people. And 80% of rare disease are genetic or have a genetic component. But yet, so far to date, the average diagnosis takes 8 years. I can imagine that a sequencing technology can really increase the likelihood of rare disease diagnosis and provide a much faster path for the diagnostics and also ending in this long odyssey for rare disease diagnostic. So now, in the end, I'm just going to have a little bit on the common disease. And one of the common analysis used for this is called genome-wide association study, GWAS. So this analysis is able to uncover variants that are associated with disease. Okay. So what happened here is that we basically have a group called cases or affected cases, basically with a disease or phenotype. And then you also have a control group, and they are unaffected controls, okay, individuals. Basically, you sequence both populations, and then through GWAS, you're able to identify that genomic variants that is statistically associated with certain risk of disease or particular trait. Okay. So important, again, you have to have two groups for this analysis. And to date, this analysis has successfully identified variants for many different types of specific complex disease. So basically, for example, type 2 diabetes, Parkinson's disease, and many more, and study still. Okay. Now, I hope I convinced you that sequencing and the variant identification is really important for human genomic research. But then next, I'm going to move on to some other applications, just to let you know there's some other ways and applications can be used through sequencing as well. So I'm going to start with the mitochondrial DNA sequencing. And as I mentioned briefly earlier, so why researchers want to study mitochondrial genomes? So actually, mitochondrial disorders is a heterogeneous disease that can arise at any stage of life. And there's a phenomenon called heteroplasmy. Basically, it means mutate mitochondrial DNA and wild type actually can coexist. And this actually causes human disease, and including cancer, heart disease, and many other type of disease as well. So studying mitochondrial genome actually has broad applications just to understand human disease, population and genetics, and biodiversity assessments as well. And importantly, studying mitochondrial DNA or genome can help us for human identification and forensic applications as well. So and at Novogene, we actually provide a specific service for this as well. And then another application now is on the rise, is pan-genome sequencing. So basically for this, researchers are not satisfied with one single reference genome anymore. So basically it looks beyond just one single reference genome. So what is a pan-genome? It's basically a total collection of genes shared by a species and including common genes, also known as core genes, and also those unique to specific individuals as well. And through pan-genome, basically you're able to capturing a full range of genetic diversity. Basically, it's error-based area all at once, you're capturing everything. Okay? Why is this important? Pan-genome can help us to understand diversity, understand adaptive strategies, and very important precision agriculture, and also understand pathogenicity insights as well. So here's some very recent discoveries from pan-genome study. We basically have revealed that there are more genetic diversity within the maize species than between human and chimpanzee. So this is quite surprising, but it's also a highlight the biodiversity that contains in maize species. So how to generate pan-genome, you may ask. So there's two key things to consider. So one is to generate equivalent quality genomes for many samples with little variability. And then it requires robust and accurate assembly. So what you do here is very simple. Just basically have a high quality genome, sequence it for many individuals, and then assemble them, and then conduct a comparison in a simple term. Basically, you will end up a pan-genome contains core genes and also genes that are specific to individuals as well. Okay. So the last application I wanted to mention is microbial sequencing, basically, and more specifically metagenome sequencing to understand microbial complexity communities in complex environment, basically. So why this is important? One of the key advantage of metagenome sequencing is it provides a culture-independent insight into microbial community. So you don't have to capture those microbes anymore, but still can understand the genomic content of the community. So generally, it have two different major approaches. One is targeted metagenome sequencing, also known as a sequence or amplicon-based approach. Basically, it's sequencing a target gene or marker gene and use this gene information to assign taxonomy, to study taxonomy, basically. Another approach known as whole metagenome sequencing or shotgun metagenome sequencing. For this approach, basically, you sequence an old DNA content obtained from the community. So this is a more comprehensive approach, obviously. Okay. So just a little bit taste on different type of applications for DNA sequencing. Okay. So and now I'm just going to give you a little bit taste about what to expect from Novogene in 2024. And so you already heard from our account manager, Amy, that our German lab is now operating and bringing the state-of-the-art genomics services closer to you, especially for our customers located in the mainland Europe. And also, PacBio rival-based service are arriving soon also in our German laboratories. And we're going to expect upcoming upgraded capacity and automation setup as well. And so this will make our service more efficient, and turnaround time can be further improved as well. And also, we're going to launch upgraded epigenome sequence services and spatial transcriptome services soon in 2024. So please stay tuned and for more to come. And so in the end, I'm just going to have a quick recap. So we talked about DNA sequencing in general, and I talked about sequencing platforms and their principles, and I did a quick comparison there. And we also discussed a different DNA sequencing approach, and then the applications of mutation detections, especially in human genome research. And then we also touched on some other important applications as well. Okay. And especially for microbial sequencing, if you want to know more, our upcoming webinar next month is on this topic. So please join us if you'd like to know more. And so here, basically, I only touch for DNA sequencing, but you can see we also provide other solutions for DNA sequencing to meet your needs, and also, of course, RNA sequencing solution. We didn't talk much today, but we do have quite a broad solutions for you to choose from for RNA as well. Okay, I think that brings the end to the main content. Yeah.
Thank you, Shichi, for that excellent comprehensive overview. Yes, we've received some interesting questions from our participants, so I'll dive into those without further ado. First question. Which platform would you suggest for genotyping? Still Illumina?
Yes, because the reason for this is because genotyping, you really focus on just calling SNVs and indels, and Illumina is still the best or short-read platforms in general because it has high accuracy, cost-effective, and yeah, so that is still the platform, the most suitable one to date.
Yes, thank you. Next question. Did I understand correctly? Long reads are suitable for the whole genome sequencing and short reads for the targeted NGS?
Not 100% correct. So for the de novo assembly, basically, of course, long read effects, especially if you're interested in large structural variants or phasing, so long read would be really suitable. And also, you can use a combined approach to use both short read and long read as well to combine the advantages. And then for target region, generally speaking, to date, most broadly used platform is still short read, but there have been some development for long read based target sequencing as well. And also for amplicon sequencing, 16S or ITS, things like that. If you have a longer fragment want to be sequenced, long read can also be used. So yeah, it's not a clear cut. It really depends on your research purpose and your experiment design. I want to address your research question, basically.
Thank you. Next question. For whole genome sequencing of unknown species, so species without reference genome, would you recommend using Illumina platform or long read sequencing technologies?
Long read or combined. Long read or combined, yeah. So short read won't give you enough for won't give you a high quality assembly if you actually want a reference quality a genome. So you will have to go for the long read. Yeah.
All right. And next question. Can you get conclusive results if you do whole genome sequencing in patients with a rare disease, but do not have access to ancestry samples? If yes, is there a suitable sample number?
Oh, this is tricky. Generally speaking, usually designs will require compare. You will have to, generally speaking, have a baseline. Basically, then you compare with a group with phenotype or disease, and then to identify what could be potentially associated with it. So it doesn't have to be ancestor, but when you come, so there's many different ways that really depends, what type of disease you study. Okay, if this generally speaking, you're expecting only one gene impact on this, or this is a common complex disease, a lot of things can contribute to it. So also that's related to the design, the population size, case size you want to use. Again, really depend what type of disease you're trying to study, okay, and what is your aim to identify here. And I would recommend to refer to published papers or literatures or industry guides on a similar type of disease. Is that usually give you a good starting point for design. Okay, yeah, so understand what you wanted to achieve is really important. And also don't forget, sequencing or DNA sequencing is not the only approach. There are also other, RNA-seq, for example, and other approach to help clinical data. Those parameters also can be helped to analyze or understand disease as well. Yeah, so I hope
Exactly.
that helped.
Okay. Thank you. Then we'll address one final question. So the final question is, when we ask for an experiment or plan a new sequencing methodology with you, will it be possible to receive a demo result file so that we can decide if that is the way or what we are exactly looking for?
Yes, generally speaking, yes. But for a lot of the applications I mentioned, we do have a demo report that can be shared with our customers or clients, and also that include, what is the analysis content, and that is included. But also, I would like to encourage and the researchers, clients yourself, to also identify, published literatures or papers that has a similar design or has a similar research goals, and then to share that with us and to help us understand what would you like to achieve. Okay, so that also help us to identify what would be the most suitable approach as well. So I think it takes both and to have a successful projects together.
Thank you. That's an excellent way to wrap this up for today. So thank you to Shichi once again, and to all of the participants. Thank you for your time today. We hope you have enjoyed the webinar and also found it useful. And thank you for all of the questions. If we haven't managed to answer your questions today live, we will get back to you via email next week with the answer. After this webinar, you'll receive a link, an email with a link to the webinar recording and a slide deck along with a short survey. We'd appreciate you taking the time to complete that survey, since it helps us ensure that the future webinars are even more relevant to you and your research goals. In the meantime, please stay in touch. Here on the slide, you can see different ways of doing so. You can, for example, follow us on social media, Facebook, LinkedIn, Twitter, or you can contact us via our website form on novogene.com. We will put you in touch with your local contact person. We look forward to seeing you on our next webinar that is briefly visible here. So until that, I wish you all a great rest of your week. Thank you.